Good afternoon to one and all present here. I, Ms. Bhavisha Patel, working as an Associate Professor, School of Pharmacy, Parul University. I feel privileged to extend my greetings to respected Dr. M.N. Patel, sir, Provost, Parul University, Dr. Abhay Dharamsi, sir, Dean, Faculty of Pharmacy, Dr. G.S. Chakrabarti, sir, Principal, Parul Institute of Pharmacy and Research, Dr. Lalit Lata Jha, Principal, School of Pharmacy, and Dr. Veena Patel, our today's resource person. She is Associate Professor at Indu Kaka Ipkowala College of Pharmacy, New Vidyanagar, and dear faculty members and my dear students. On behalf of Faculty of Pharmacy, Parul University, I welcome you all, the distinguished dignitary, to grace the online national level webinar on regulatory environment of herbal drug industry, Indian perspective. So, wholeheartedly welcome, Dr. Veena ma'am.
Thank you, madam.
Okay. Now it's my great pleasure to introduce Dr. Veena Patel. Madam has received Outstanding Local Branch Award by Indian Pharmaceutical Association, Mumbai. She is serving as an Honorary Secretary of IPA Anand Local Branch for the year 2016, 2018 and 2020. She has received total research grant fetch of rupees 4,70,000 from state and national agencies. She has organized several seminars, conferences, refresher course as a coordinator, and served as a resource person in a state and national level seminars. She has completed more than 10 institutional consultancy projects. She has also published total 12 research papers in a peer-reviewed national and international journals, and also presented 9 research papers in a national and international conferences. So again, I am welcome to Dr. Veena madam, and thanks for accepting our invitation. Now over to you, ma'am.
Thank you, Bhavisha madam, for kind introduction. Very good afternoon to one and all present over here. First of all, I am very much thankful to management of Parul University for giving me this opportunity, as well as Dr. Lalit Lata Jha madam, principal, and obviously Bhavisha madam, and Dr. Anand Pichadia sir, vice principal of Parul School of Pharmacy, Parul University. So today I have selected a topic is regulatory environment of herbal drug industry, and here today I'm here to present my view on the topic with Indian perspective. So we are knowing that nowadays people are moving towards herbs, and according to World Health Organization, 80% of population of developing countries, they rely on herbal remedies as a preliminary healthcare. Okay, and nowadays developed countries are also showing their interest in herbal. Okay, so with this reference I have selected this topic, and before starting the topic, we just need to go through several, this is the outline of presentation. I have included several definition, and is necessary to understand the various terms which are going to be used in throughout the entire presentation. Then we'll discuss about the scientific validation of herbal medicine in terms of quality, safety and efficacy. Then we'll discuss about stability testing for herbal medicines. Then regulatory aspects of Indian traditional medicines with Indian perspective. Then we are also looking for international scenario of in terms of regulatory aspects. Then we'll discuss about India and traditional knowledge, that is known as TK, biopiracy of TK, and success of traditional knowledge digital library, that is TKDL, against biopiracy. So here we start with several terms that are necessary to understand, as students are also there in this presentation. Okay, so first of all, we must know what is considered as herbal medicine. Okay, so herbal medicine, that includes whether it is a herb, herbal material, herbal preparation, finished or and finished herbal product. So anything can be considered as herbal medicine. Okay, so it is not like that you'll be having some formulation, ready formulation in your hand, and then it is considered as herbal medicine. As we are Indian, and since our long time we are using herbal medicine, so we are using in any form, like herb can be used, herbal material can be used, herbal preparations are also used, as well as finished herbal products. Now herbs include, let's see what are the herbs, what are herbal materials, and likewise, herbs include crude material which could be derived from lichen, algae, fungi, or higher plants such as leaves, flowers, fruiting bodies, seeds, stem, wood, bark, root, rhizome, or other part, which may be entire, fragmented, or powdered. Okay, so though anything can be considered as herb, so it would be related to the plant. So whatever plant part is there, that is considered as herb. Either it is whether it is entire form or fragmented or a powder form. Then herbal material that include addition to the herb. Okay, it includes herbs, whatever criteria we had seen under the category of herb that are included in herbal material. Apart from that, it also include fresh juice, gum, fixed oil, resin, dry powder of the herb. In some of the countries, this material may be processed by various local procedures such as steaming, roasting, stir baking with honey, alcoholic beverages, or other material. So that is considered as herbal material apart from herb. Now herbal preparations. Okay, so some we are going to prepare, or we are going to process herb or herbal material to get the herbal preparation. Okay, so these are the basis for finished herbal products. So whenever you want to make any finished herbal finished product, so you need to require, you must have in herb in form of herbal preparation. Okay, so it is optional, you can use herb or herbal material also, but herbal preparation is most of the time it is a basis for finished herbal product, and may include comminuted or cut herbal material, or extract, tinctures, and fatty oil of herbal material. They are produced by extraction, fractionation, purification, concentration, or other physical or biological processes. They also include preparation made by steeping or heating herbal material in alcoholic beverages and or honey, or in other materials. So everything can be considered as herbal preparation. Okay, now last is finished herbal product. So it consists of herbal preparation made from one or more herb. If more than one herb is used, the term mixture herbal product can also be used. Finished herbal product and mixture herbal product may contain excipients in addition to active ingredients. Okay, so whatever active ingredients, here we are not going to add isolated constituent. Okay, it is clearly mentioned in the guideline given by WHO that we are not going to add isolated constituent or any synthetic compound which is a pure compound, like that we are using in conventional dosage form, we are using API. So it is not there with herbal one. So it is mentioned in the definition itself: if a finished herbal product or mixture herbal product to which chemically defined active substance have been added, including synthetic compound and or isolated constituent from herbal material, are not considered to be herbal. Okay, so even if we are isolating quinine from the cinchona bark, and then that isolated quinine we are adding, even though is not considered as finished herbal product. Okay, so we just have to use any herbal preparation that we had seen in the previous definition. Okay, now for the quantification as well as for the standardization purpose, as we are going to deal with the regulatory guidelines. So for the standardization of a herbal material, marker compound is necessary. Okay, so for the, in case of conventional dosage form, we are using reference standard. So here we are using marker in case of herbals. So marker are chemically defined constituent of a herbal material utilized for control purposes. They may or may not contribute to clinical efficacy. when they contribute to the clinical efficacy, evidence that they are solely responsible for clinical efficacy may or may not be available. Okay, so it is not like that whatever marker you are using, that is only solely responsible for particular therapeutic effect or efficacy, right? Sometimes we are using the marker compound which is not having said therapeutic activity for which the product has been used. Marker are generally employed when constituent of known therapeutic activity are not known or are not clearly identified and may be used to identify the herbal material or preparation or calculate their quantity in finished product. Okay, so for the quantitative purpose, we are majorly we are using the marker compound. Now, this all these term that we had discussed, that is about herbs. Okay, now our traditional system of medicine that existing in India, and one of the famous system is Ayurvedic system. So let's see what is the definition or how it differs from the herbal one. So Ayurvedic drug that includes all medicine intended for internal or external use for or in the diagnosis, treatment, mitigation, or prevention of disease or disorder in human beings or animals, and manufactured exclusively in accordance with the formula described in authoritative text of Ayurvedic system of medicine and Ayurvedic Pharmacopoeia of India, specified in first schedule of the Drug and Cosmetic Act 1940, rules 1945. Okay, so what is the major difference? We need to strictly follow the procedure. Okay, in case of Ayurvedic drug, whatever Ayurvedic medicines are there, they are strictly following the procedure whatever mentioned in the ancient text. Okay, so that we can refer any authoritative text of Ayurvedic system of medicine, or we can refer even Ayurvedic pharmacopoeia. So that is about Ayurvedic drugs. Okay, now let's start with the scientific validation of herbal medicine. Okay, so whenever we are giving some of the product to the market, to the society. Okay, so it must be scientifically validated, and for that several parameters need to be studied, and we need to provide some standardization reports also. Okay, so that's why it is mandatory as per the guideline. Now it is I have divided it into three major part. First is quality of herbal medicine. Okay, so for the quality, we are going to study quality evaluation of herbal medicine and whatever constraints, challenges, and opportunities are there that we are going to discuss. Then second is safety of herbal drug, and next is efficacy of herbal drug as a part of scientific validation. Now, first, we'll discuss the quality evaluation of herbal medicine. That includes whatever factors and affect the quality of herbal drug. Okay, that are factors affecting, first of all, factors affecting quality of herb, then adulteration of herbal drug, deterioration of herbal drug, counterfeiting of herbal drug, and lastly, we'll discuss several evaluation methods for the herbal drug. So first, let's discuss the factors that affect the quality of herb. Okay, so if we want to make persistent quality of herbal product, okay, then we need whatever raw material we are having, it should be having uniformity in terms of quality. Okay, and there are number of factors which affect the quality of raw material. If you are not having a proper quality raw material, then we cannot give the proper quality finished product. Okay, so this is the obvious thing. So several factors which affect the quality of herbs, and that are authentication and reproducibility of herbal ingredient. So number of times people are not going for the authentication, and in Gujarat we are having one proverb that bar gaave boli badlai. Okay, so that means one of the drug is identified by the name in Vadodara district. So the same drug is identified by the another name in Anand district. Okay, so authentication, and that's why authentication is mandatory. Whatever drug is identified by common name, so you must have to authentify the raw material by its botanical name with the help of botanist. Okay, and then you have to go further. So authentication and reproducibility of herbal ingredient, it is necessary. Inter and intra species variation in the plant. Okay, some of the most of the times what will happen, when we are dealing with the plant material, they are having different species existing in nature, and without checking what species is mandatory for the collection, people are using whatever species is available at a time, right? So it also affect the quality of the drug. Then several environmental factors are also affecting the quality of raw material, herbal raw material, like solar radiation, soil nutrients, different kinds of stress, seasonal variation. Okay, so whatever season you are selecting for the collection of the medicinal plant, so it should be collected in a proper season, right? Like if you are collecting, if it has been mentioned, like you have to collect the medicinal plant when there is a flowering tops are there, right? And before flowering tops, if you are going to collect the plant material, so there are chances that whatever active compound is present, it would be present in the plant that we are not getting in enough amount. Then geographical location is also affecting, one of the best example is of cinchona bark. Cinchona bark is widely used for isolation of the quinine. Okay, so if the cinchona plant has been cultivated in Himalaya region, they are very well grown in Himalaya region, and they are very good in terms of quinine content also. Okay, but the same cinchona plant, if you are cultivating at a sea level, so what will happen? They are cultivating, or they are growing very well as that of the plant growing in a Himalaya region, but this plant are devoid of quinine. Okay, so for whatever purpose you have cultivated cinchona plant, by changing the geographical location, and your purpose is not solved. Okay, so we need to check the geographical location also, plant parts used. Okay, so what your part you have to use is in case of clove flower bud. Okay, they are having flower bud, clove, lavang or loving. They are having its a shape in that is having a stalk like structure and crown like structure. Okay, and further stem part is also having the brown color, but is having less amount of volatile oil. Okay, so we need to take care that what plant part we are going to use, time of harvesting, whatever proper season or proper time is there, it should be followed. Post harvesting factors like collection, preparation, drying of the plant material. Okay, which type of drying method you are following, so these all are post harvesting factors. Then contaminants in herbal ingredient, environmental contaminants, pesticide, fumigants, and other toxic metals. So these are also affecting the quality of herbs. Like nowadays, just to make easy and easygoing, so people are using pesticides and fumigants in a quantity. Okay, they are not taking care about. They are not having any concern that these pesticides are going to absorb by the plant, and they are going to retain in the soil also. So whenever we are going for the next crop, so even though that pesticide has not been used for the second crop, then also they are found to be present in the next crop. Okay, so the use of pesticide, fumigants or herbicide or synthetic fertilizer should be avoided, or it should be used in some limit. Okay, so this should be fixed by the authorities, or it should be mentioned in the guideline also. Chemical variation in medicinal plants. So some as is a natural process, chemical variation. So if we are not checking before utilizing that herb into the medicine, into the our formulation, so there are chances that it will affect the quality, because there are possibilities that that chemical compound is totally absent, or it is present in too much less quantity. Okay, so we must need to check chemical compound which are present in medicinal plant when it is in a form of raw material. Now, second point for the scientific validation, adulteration of herbal drugs. So whatever herbal drug we are using, herbal material that we are using for the preparation of finished herbal product, so you have to go for the check that whether any adulteration is there or not. So what is adulteration? Adulteration or debasement of an article refers to a set of deliberate or accidental admixture in which undesirable materials are admixed with herbal medicinal product. Okay, so deliberately, that means knowingly or unknowingly, people are mixing the similar looking substance or totally different plant material they are giving. Okay, and that causes the adulteration. Now there are several means listed over here, like substitution with standard commercial variety. Okay, so whatever standard variety is there, that is rich in terms of active phytoconstituent. Okay, and if that variety is not available, then somewhat inferior variety. Okay, inferior in terms of its quality, otherwise it is having similar morphological characteristic, as well as it is also showing presence of active phytoconstituent, but might be they are in a lesser amount. So that is called what? Substitution with standard commercial variety. Then substitution with superficially similar but inferior drug. Superficially similar, that means મોર્ફોલોજીકલી they are same, બહારથી આપણે જોઈશું તો બેઉ સેમ દેખાશે, પણ inferior drug, that means what? Is inferior in quality, that means is not having active constituent present at all. Okay, so if active constituents are not present, then is not going to give you a therapeutic effect. Okay, so that is the second case. Then substitution with artificially manufactured substance. Okay, so the best example is chicory. Chicory is artificially manufactured, and it has been adulterated with the coffee beans. Okay, so if you read the labels of coffee powder which is available in the market, you will find that they are mentioning also that they were added chicory. Okay, so that this is artificially manufactured substance example. Then substance with exhausted drug. So this is the case most of the times with volatile oil containing drug. So volatile oil containing drugs, in that volatile oil has been isolated, and whatever drug is remaining, whatever plant material is remaining, they are dried again. Okay, and if required, they are going to colored also, and they are sprinkled with artificial flavor also. Okay, and then they are again sent them into the market. So these are the totally exhausted drug. Okay, so whatever volatile oil containing drugs like fennel, coriander, clove, cardamom. So it is possible that whatever sample is available to you, it is exhausted. Okay, so you need to identify whether it is adulterant or not before utilizing them into the process of manufacture of a herbal product. Use of synthetic chemicals. Some of the natural plant material like balsam of Tolu, balsam of Peru, they are having naturally they are having the smell of benzaldehyde. Okay, so benzyl benzoate, that is the chemical which is used as an adulterant. Okay, and just assume it is how much risky. Then next is presence of vegetative matter from the same plant. If you require a stem part, then definitely all the raw material is having only and only stem part. It should not be having any root part, or it should not be having any leaf material. Okay, so if along with leaf you are getting some pieces of stem, then stem is considered as adulterant, as it is a vegetative matter of same plant. The next is harmful adulterant. One of the example is colophony. Is a crude drug. So colophony is having a appearance like amber colored broken glass pieces. Okay, so what people are doing, they are mixing the broken pieces of amber colored glass. Okay, and again is considered as what? Harmful adulterant. Another example is rodent fecal matter is added in cardamom seed. Okay, so whatever cardamom seeds we are having, they are just having a shape and appearance like that of rodent fecal matter. So when we are so it's my personal advice that never purchase what cardamom seeds. It's better to go for purchasing of cardamom fruit. બહુ તો એની અંદર exhausted હશે, બરાબર, but we are not getting any animal fecal matter. The next is adulteration of powder. So whenever we are purchasing powder instead of crude drugs, so it is very difficult it is very easy for the suppliers that what they are doing, they are mixing the similar colored powder. Okay, so that is the another type. So adulteration of herbal drug, it should be one of the parameter of validation. Okay, now deterioration of herbal drug. So we must have to follow the guideline for the good agricultural practices. Okay, then good agricultural and collection practices, this kind of guidelines are available. So in that they have clearly mentioned that how to process herbal drug. So once they are collected, okay, and how to process and how to store. Okay, so storage is also important characteristic in terms of in case of herbal material, as all the herbal material, if they are not properly dried and if they are not properly stored, so there are chances of microbial contamination. Okay, and once it had been contaminated with this microorganism, with some kind of bacteria or fungi, then you cannot use that kind of raw material for further process. Okay, so herbal drug, which is there are chances of deterioration of herbal drug by light, moisture, temperature, air oxidation, bacteria and mold, mites, nematodes, worms, insect and moths. Okay, so we must need to go for the proper storage as well as proper drying of herbal material to just to prevent it from deterioration. Now, counterfeiting of herbal drug. If you are not having any source of right source, whatever you are looking for, that material is not available to you, and you, instead of them, you're using some another material. So that is counterfeiting. So counterfeit drug that contain same dose of active ingredient. Okay, so drug is different, but it contains what same dose of the active ingredient. It might be present in some another plant. We know that most of the class, like for example, alkaloids, glycosides. So number of plants are showing presence of same alkaloid, same glycoside. Okay, so it is not like that aloin is present only in aloe drug, right? It is also found to be present in aloe barbeloin is also present in senna, right? So in senna or in rhubarb. So one compound might be present in another drug also. So what people are doing, they are using the same totally different plant. Then counterfeit drug that also include mislabeled medication. It is not labeled properly. Then counterfeit drug that contain incorrect dose of active ingredient. Incorrect dose. Okay, so whatever dose it should be given to the patient, or it should be used by the prescription of any practitioner. So dose is totally incorrect. It should counterfeit drug that do not contain the active ingredient at all. So just like placebo drug available in the market. Counterfeit drug that that containing potentially harmful substances, that is unlisted active ingredient. Then genuine medicine marketed for the incorrect or recreational uses. So these all are possibilities of counterfeiting drug, and it should be completely regulated. Okay, complete regulation is required for traditional herbal medicine to just as a precaution from the counter utilization of counterfeit drug. Now, these are the evaluation methods of herbal drug. Okay, so mainly whenever you are using any herbal raw material, you want to use herbal material for the making of any herbal product or even any traditional system of medicine that include Ayurveda, Siddha, Unani product, right? So ASU medicines are also utilizing one of the sources plant material, right? So whenever you are dealing with any plant based raw materials, so these are the several evaluation methods. So first all, there are mainly 5 methods: botanical, physical, chemical, biological, organoleptic. Okay, so first we'll start with the organoleptic. Whatever like color, order, taste, texture, fracture. Okay, so whatever we see it, whatever we taste it, okay, whatever we feel it, that all are organoleptic evaluation parameter. Then botanical parameter, that are that is divided into 2 part: macroscopic and microscopic. Okay, in that shape, external appearance than any marking. In case of microscopic qualitative microscopy, you can go for it. You can go for the quantitative scanning, electron microscope studies, powder studies. Then in case of physical, moisture content, extractive value, ash value, fluorescence analysis. In case of chemical analysis, you can go for the qualitative, quantitative by using sophisticated instruments like fingerprinting, okay, and you can find out the presence of secondary metabolites also. You can go for the different chromatographic techniques. You can go for the pesticide residue, mycotoxin, whether it is present in the raw material or not. And last is biological study. In that, you can go for the microbial contamination. You can go for the pharmacological uh, activity of a particular raw material. Toxicological studies are also performed. Okay, and any other specific activities. Okay, so that is decided by for which purpose and what kind of material you are using. So this is about all scientific validation of herbal material. Now, safety of herbal drug. Okay, so safety, as we know that commonly people are using the word that if you ask that why you are preferring herbal drug, the one common answer will be getting is what they are having no side effect. Okay, so it is not at all correct. Statement that they are devoid of any side effect. They are having side effect, they are having toxic effects. Okay, so safety of herbal drugs. And another thing in our mind is what that herbal medicines are timely tested. Okay, and, since last so many years people are using herbal drugs, so they are not getting any adverse effects, so why we? Okay, so this is the common thing and is not true because nowadays since so many years, and as we discussed that people are using pesticides, they are using synthetic fertilizer, fungicides are used, weedicides are using. Okay, so the residual effect is always present in the plant material, and previously it was not there. So there are chances that due to high amount of residue, pesticide residue, it may create toxic effect. Okay, due to high amount of heavy metal which is present in the plant material, so it also create toxic effect up to the mortality also. It may create mortality also if it is having some high amount of heavy metal. Then plant material which is having a history of aflatoxin. Okay, so you cannot use that kind of plant material which are having without tasting. Okay, so safety is also having concern before utilizing the herbal material. So in that we are going to discuss about the pharmacological and herbal medicine, pharmacovigilance system and safety monitoring of herbal medicine, sources of adverse drug report of herbal medicine, specific area for safety monitoring of herbal product. So first definition of pharmacovigilance. You are very much aware with the pharmacovigilance system that it is the science and activities relating to detection, assessment, understanding, and prevention of adverse effect of drug or any other possible drug related problem. Now, pharmacovigilance that covers several issues like substandard medicine, medication error, lack of efficacy report, medicines having inadequate scientific basis, acute and chronic poisoning, drug-related mortality, abuse and misuse of medicine, adverse interactions. Now, pharmacovigilance is working with the aim improve patient care and safety, assessment of benefit, harm, effectiveness, and risk of medicine, encouraging their safe and rational and more effective use. Okay, and to achieve this aim, uh, by these aims are achieved by early detection of previously unknown adverse reaction and interaction, identification of risk factor and possible mechanism, estimation of the benefit, risk, and analysis. Now, pharmacovigilance system and safety monitoring of herbal drug. So, as we discussed that herbal drugs are widely used, they are widely accepted, they are concomitantly high consumption is there for the herbal drug. And one more thing that they are taken with other medicine. Okay, people just they are having mindset of like herbal drug can be used at any time and with any kind of medicine. Okay, so they are on the conventional uh, treatment, conventional medicinal treatment. So even though without informing the practitioner, they start to take herbal medicine. Okay, so these are the facts about herbal drug. Now, monitoring of safety of herbal medicine that requires for the benefit of public health. Okay, so it should be in the benefit of public health as well as individual health, to identify the risk associated with their use, to assess and understand the problem of any adverse effect arising. So with that, pharmacovigilance is working. Now, sources of adverse drug report of herbal medicine. Okay, so finding the information about adverse drug reaction or adverse effect of the any used herbal medicine. So first source is healthcare professionals. Okay, so they are directly in contact with patient care, and they are directly dealing with the patients. Okay, so they are the great source of this kind of information. If any adverse drug reaction has been reported or has been known to them, so they need to report further. Then we know that these herbal medicines are what they are, non-prescription medicine. Most of the drugs are easily available, right? No, they are non-prescription medicine, and some of the prescription medicines also, but they are easily available. Okay, and so another source for reporting of adverse drug report is um, community pharmacist and nurses. Okay. Then another source is manufacturer. Like they can also call the information, collect the information from the consumer and healthcare professionals. Okay, so whatever drug they are formulating, okay, and for that they can ask the information from the healthcare professionals, from the hospitals, okay, by sending their representative, and in this way they are collecting the data. And other sources are also there, like National Poison Center, then Drug Information Center, and Consumer Organization. So these are the different sources that are collecting the information, or we can find some adverse drug reaction information from these people. Now, specific area for safety monitoring of herbal product. Now, safety monitoring, we just divide the herbal products into two categories. Okay, according to their regulatory status, and the second is according to their registration or marketing status. So, according to their regulatory status, whether if the drug is under prescription medicine category, non-prescription medicine category, or any other herbal product. Okay, so according to their regulatory status, you can find out specific area. While for the registration and marketing status, new drug develop any herbal drug which is under new drug development process. Okay, so at the time in clinical trial prior to national drug regulatory approval. Then second is under post marketing safety surveillance, when the herbal drug is under new drug development process. So these are the two possibilities that we can get the information. Then herbal drug under re-evaluation of the current protocol. So again, the drug, if it is in a clinical trial, or if the drug under post marketing safety surveillance. Then herbal medicines on the market, which are available in the market, and that under post marketing safety surveillance. Then other herbal product marketed for the healthcare, such as dietary supplements. Okay, nowadays it is also in great demand, nutraceuticals and dietary supplements. So they are comes under the other category of herbal product which are marketed for the healthcare. So these are the two different areas or possibilities where we are getting the specific safety monitoring of, we can keep our eyes watching on safety monitoring of herbal products. Okay, so once we had checked its safety, we had checked its scientific validation data. That means we had checked its quality. Okay, quality is perfect. Then it if it is proven to be safe, okay, then go for the efficacy of herbal drug, how much is efficacious. So for that, there are two widely used models are there: in vitro screening models and in vivo screening models. Okay, so first let's discuss in vitro screening models. Plant may have evolved over a period of time, leading to changes in their chemical composition and thus the biological activity. Okay, so as we discussed, like whatever herbal material available in before 2000 years. Okay, so the same herbal material is available in these days also, but there is a change in composition of active compound or any chemical compound, right? Whether it is active or inactive. So that is the changes due to number of factors are affecting, like environmental factor and using of the synthetic and all that. Okay, so there are change in their biological activity. So that's why it is necessary to establish the therapeutic efficacy of medicinal plant, to confirm their historical utilization. Advantages of in vitro screening: one can easily study and compare the efficacy of different plant in a cost effective manner and design rational drug combination. Two approaches are used, like screening multiple plant using a single bioassay and studying the action of single plant with multiple bioassay. So these are the two approaches for in vitro screening. There are several limitations with in vitro screening, like the lack of positive result. Okay, sometimes we are not getting positive result with in vitro models. Okay, so it's not like that is the plant material is devoid of active constituent. There are several reasons to get this negative result. Negative result are uh, the assay employed may not be appropriate. Okay, whatever assay method you selected is not appropriate. In vitro assay do not screen for active principles that are generated as metabolized product in vivo. Okay, so initially that compound, that plant, that active compound is present in plant material is going to metabolize once it has been taken orally or given to the patient. So it is metabolized and then is uh, comes into its active form. Okay, and then is going to give some therapeutic effect. The crude extract may contain insufficient quantity of active component, resulting in extract being inefficient at the dose tested. So, like we are not knowing that in what amount the active compound are present in particular dose. So, if we are making some mistake in selection of dose, then again we are not getting proper result. Now, second is in vivo screening, that is divided into two parts again: preclinical and clinical studies. So, preclinical screening of botanical involves documentation and testing of their pharmacological efficacy in vivo models and studies of toxicology, specificity, biopharmaceutical properties, and drug interactions. Why it is necessary? Some extract may show in vivo activity due to metabolism of inactive compound into active form. Okay, as we had seen in earlier slide, can help in evaluating efficacy and safety data before performing clinical trials. Negligence of toxicological studies on herbals. Okay, so as we just presume that it is not toxic and not going to go for the toxicological studies. Okay, so the belief that it's prolonged and apparently uneventful utilization is an indication of its safety. Okay, આટલા બધા ટાઈમથી બધા લોકો લે છે, so is not a toxic, so you can use it without performing any toxicological study. Traditional practitioner may not be able to monitor delayed effect, adverse and rare adverse effect due to long-term use of this plant. Importance of toxicological studies. its study should be include tests such as acute, chronic, subchronic, and special toxicology that are impossible to detect clinically, such as genotoxicity, immunotoxicity, reproductive toxicity, and carcinogenicity. Okay, so this about this kind of toxicity we are not getting idea from in vitro models. So it should be performed in vivo, and this kind of models should be selected and it should be performed. This test help in identification of toxic symptoms and possible organ involved also. Special toxicological assays such as carcinogenesis are required if known mutagenic or carcinogenic compound are present in the plant. So if the plant is having a history of this kind of compound, it must be tested whether your plant material is having that compound present or not. Now, next is pharmacokinetic and bioavailability. Pharmacokinetic can be used to link in vitro assay and clinical studies. It can help in obtaining the information on bioavailability of active constituents. Bioavailability often determines the therapeutic efficacy on that product of that product. Pharmacokinetics can be used to study herb drug interaction. For example, tamarind and aspirin. Okay, so tamarind is widely used in our Indian kitchen and is also one of the ingredient of Ayurvedic formulation, right? So when a tamarind is taken by the patient and is given aspirin of 600 milligram single dose of 600 milligram aspirin, then it will increase the absorption of aspirin. Okay, if tamarind is taken, so it will increase the absorption of aspirin and may cause severe toxic effect also. It helps in designing safe and rational dosage regime. Now, second part is of in vivo method, that is clinical trials. Once you are having all the data set of preclinical studies, that including in vivo models and toxicity studies, everything has been performed, then go for the second part, that is clinical trial. One of the major issue in modernizing traditional medicine is measurement of its efficacy. User surveys, case studies, and anecdotal information regarding traditional medicine are not accepted by many clinical practitioners. બરાબર, તમે એવું કહી દો કે આટલા બધા યુઝ કરતા હતા, આટલા બધા લોકોએ યુઝ કર્યું છે, આટલી ઇન્ફોર્મેશન અવેલેબલ છે, then if you want to sell your product in some developed countries, in some European countries, then they are not going to accept this thing, right? કે તમે આવા verbal communication કે કોઈ જેનો proof નથી એવો data, right? So they claim that without clinical trial, the efficacy and safety of herbal medicine cannot be demonstrated, and insist on implementation of method and guidelines used for clinical validation of modern medicine on herbal products. Randomized control trial are widely used. Placebo randomized control trial, that is placebo RCT, which is considered the gold standard, are more difficult to undertake due to the requirement of designing suitable placebo. Okay, so these are some limitations with the herbal drug, that we are finding difficulty in designing some placebo models, right? Development of a method to allow placebo control for changing and individualizing therapies is required to facilitate comparison between traditional medicine system and Western medicine. Considering the fact that herbal medicine is individualized and offer a holistic approach, adaptive clinical trial to retain the therapeutic relationship is required. Now, so that is about our validation of herbal product. Okay, now second point is stability testing of herbal medicine. So stability is a big issue with the herbal medicine, that we know that number of factors are affecting during manufacturing, during transportation, and during storage of the herbal material. That several it may, if it is herbal product is not remain stable. Okay, so people are not having faith in herbal formulation due to the stability issues, and this is also one of the reason that is not widely accepted by developed countries. So drug regulatory agencies across the globe have recommended guideline for the conduct of stability studies on herbal drug product, which require that stability data should be included in a product registration dossier. Okay, so if you want to go for marketing of your product in some other countries, and according to their guideline, you need to produce this kind of data, okay, in a product registration dossier. From the scientific viewpoint, numerous chemical constituents in herbal drug are liable to varied chemical reaction under the influence of different conditions during its shelf life. This reaction can lead to altered chemical composition of herbal drug product, and consequently alter the therapeutic profile. Okay, so during it's not like that. Some of the time it may be changing. Chemical compounds are going to change continuously, and uh, it may affect its therapeutic value. Now, there are possible interaction among the different types of phytoconstituent as we are mixing. If even if we are having a single herb formulation, right? Even though it is having a number of chemical compound present in a single drug, and if you are mixing more than one drug, then number of constituents are going to be increased. Okay, so there are several interactions are going on. For example, if some of the compound is having tannin and glycoside, okay, so water solubility of tannin is going to be increased in presence of water soluble glycosides by the non-covalent interactions. Okay, now if tannin and alkaloids, okay, so tannin will increase the precipitation of alkaloids. Then this polysaccharides, tannins, and lignins, when they react with the heavy metal, they are going to form some complex reactions. Okay, so this kind of reactions are going on, and they also affect the stability of product. Now, once we had seen that stability of any herbal product is it should be a very important criteria, and it should be evaluated on the basis of four different methods. You can go for four different ways are there, okay, to check whether it is stable or not. Okay, so first of them is a physical study. Okay, you can check whether it is physically stable or not. Okay, so for example, here in one of the study, fenugreek seed powder in form of fast disintegrating tablet, they have used this seed powder, and is going for the accelerated stability study for 3 months. So the result found that it was remain stable, but a number of times we had observed that in a herbal formulation, the stability issues, physical stability issues are big major. Like a number of times we had observed, like color of the formulation is going to change. Okay, so that is the example of physical instability of drug. Then one can go for the physicochemical studies. In that, for example, curcuma longa extract, it was tested for long-term stability at 4 degrees centigrade. So the result found significantly degradation of curcuminoids. So is a active compound, is a chemical compound, is going to degrade after in a stability testing study. Then biological studies, Echinacea purpurea formulation, if it had been tested at 430 and 40 degrees centigrade for 6 months, then in case of biological studies, like total phenolic compound and antioxidant activity is found to be decreased with stability testing. Then another way to go for the stability testing is physicochemical along with biological studies. Okay, so Azadirachta indica oil that containing formulation is tested at varied temperature from 5 to 54.5 degrees centigrade for 14 days. So here they found azadirachtin A content decreases significantly. Okay, but its a therapeutic effect was remain as such, and the chemically azadirachtin A was found to be degraded. Now, key challenges for stability testing of herbal drugs are chemical complexity and biochemical composition, variability and raw materials, selection of marker compound, and influence of enzyme. As enzymes are present in some of the plant materials, so continuously they are going to for the chain of reaction, and they are going to change the some chemical compounds also, form of chemical compounds. Now we are moving to the regulatory aspects of Indian traditional medicine. Okay, so here in India we are following Drug and Cosmetic Act 1940 and its rule 1945. Is time doesn't permit, so we are skipping the objective of DNC Act. Now regulatory aspect of India, herbal medicines are very common and they are generally used. Okay, regulation on herbal medicine product are necessary. Everything is fine. Okay, so drug regulatory authorities in India, regulatory bodies and organizations like Ministry of Ayush and Cirisco contribute a critical role in fulfilling the necessities of authorized procedure for pharmaceutical research process. So, if you want to go for the manufacturing of any herbal medicine, that is herbal or ASU medicine, Ayurvedic, Siddha, Unani medicine in India, so there is the role of Ministry of Ayush and Ministry of Health and Family Welfare. Okay, so here definition what covers the ASU medicine is given. Now for the manufacturing license, you can go for application with a prescribed form number and prescribed fees. Okay, application has been filed, then for the manufacturing, then for the renewal of your certificate, you need to go for the submit the application along with form number 26D. Okay, and once you get the license, once you are fulfilling all the requirements of GMP for ASU drug, okay, so, and then you'll be getting a license. Okay, so once license has been issued, so you can use it for the 5 years. And after getting license, licensee should maintain proper record. They should allow inspector to inspect the premises. Okay, then for the labeling, packaging and limit of alcohol for the ASU drug, label should be in English and Hindi, both the languages, and content label should be contain this many information. Okay, if it has been used for the external use, it should be written in a caps lock for external use only. Now Schedule T requirement. Okay, so you just go for the Schedule T, and you will find that these are the requirements of Schedule T. It should be also fulfilled. Then documents required. Okay, whenever any inspectors come to the manufacturing unit, okay, so at the time as such, licensee need to maintain the listed documents. Okay. Then drug information, what kind of information need to disclosed. So this is the list, like name of the product, formula shall contain Sastric Tibbi name, detailed method of the preparation, purification of drug wherever required, indication, dosage, schedule in detail, side effect, antidoses, then diet restriction if any. Okay, so this is about the Indian regulation. Now, as we discussed that European countries, okay, European countries are having strict rules and regulation regarding herbal medicine. So I'm just comparing the regulation of India with the one of the best regulation of European Union, EU. Okay, so in that, these are the requirements. Here, the product is identified by traditional herbal medicine, and regulatory agencies European Medicines Agency, EMA. And you need to submit the format for herbal medicine under common technical document. Okay, so common technical document, in that format you need to submit your proposal. And regulatory pathways, you need to select from any one of this. The definition has been given medicine. Now application requirements, so these are the list of requirement for the application under EU. Test result you need to submit. Yes, they have clearly mentioned that pharmaceutical test, preclinical test that also include toxicological and pharmacological test. Okay, so here it is compulsory. Then clinical trials, summary of product characterization, that is mentioned. Okay, and these are the detail of common technical document format, CTD format. Okay, so in this format, one has to submit the application. And this is the comparison between India and European Union. So classification, regulation, and mode of submission. Here electronic submission is acceptable without any, they are not paying any charges. Approval timeline, it requires 3 months for India. Here it requires 210 days. Okay, and both of the countries are giving 5 years of validity. Now let's quickly see the India and traditional knowledge. India and traditional knowledge. We are rich in terms of biodiversity, and we are having very rich source of ancient scriptures like Veda, Puran, Upanishad, Brahma Sutra, etc. Okay, and in the entire world, total 26 agroclimate zone has been declared. Okay, and out of 26 in India alone, we are having 16 agroclimate zone. Okay, and we are having wide variety of herb, shrub, around 47,000 of plant species which are identified. Okay, so which are apart from there, it is also number of plant species are available, but this many number is identified. And these species are used by a traditional system of medicine, that is Ayurveda, Siddha, Unani, and homeopathy. Now piracy is the word that is defined as unauthorized publication or reproduction of another person's work or material without permission. Okay, and biopiracy is what it can be defined as somewhat stealing of biomedical knowledge from communities and individuals without their consent. Okay, so biopiracy for traditional knowledge, it is the very biggest issue. Like we are, as we had seen, we are rich in terms of biodiversity, we are rich in terms of wealth of natural resources, in terms of not only plants but animals also. Okay, so we are having a traditional knowledge TK. Okay, so it is stolen by the some of the other authorities, and they are going for the patenting of traditional knowledge of India. Okay, so there is a one case, biopiracy of traditional knowledge of neem case in European Patent Office in 1994. They have granted a patent for method for controlling fungi on plant by aid of hydrophobic extracted neem oil. Okay, so in India we are using neem as so many years, since last so many years, as a fungicide. It had been used in our cultivation. So several NGOs and Indian farmers opposed this patent in 1995, and at last, after having all the arguments, they need to revoke this patent given by the European Patent Office in 1999. Okay, so it will take 6 years to cancel that. Just to explain them, this is our traditional knowledge. You cannot use. There is nothing invention kind of thing is there, and it is a prior art. It is known to public domain. Okay, so you cannot use this thing. Okay, so and out on not only 6 years, but it also requires thousands of millions of dollars. Okay, so this is about the neem case. Second case was turmeric, same way. As we know that turmeric in India we are using as a wound healing since last so many years. And it has also been mentioned in our ancient scripts in Sanskrit language that you can use turmeric as a wound healing. So how they can use, and they can go for the patent. Okay, so they can use it definitely, but they cannot go for the patenting of this invention, this use of turmeric. So again, they need to cancel the patent, which is argued by CSIR, Council of Scientific and Industrial Research. And after that, India has decided all this for Ayush. They formed an interdisciplinary task force in 1999, and they have established traditional knowledge digital library. Okay, aim of this TKDL is what? Make Indian medicine system available in public domain. If it is not in public domain, anyone can use it, anyone can steal it, and they just, as it is easily available, right? Traditional knowledge is easily available. કે તમે કોઈને બી પૂછી લેશો તો તમને કંઈ પણ information મળી જશે. But if it is in a public domain, so you cannot claim it for the patent. Okay, so now what TKDL, that is Traditional Knowledge Digital Library, has done, they have created knowledge transfer. They have provided knowledge transfer platform. Okay, so our ancient scriptures are in Sanskrit, in Arabic, in Persian, in Urdu, and in Tamil. Okay, so they have translated into the English, Spanish, German, French, and Japanese. Okay, so any patent office, they can search the TKDL, and they can convert the information in their own language, and they'll be having idea that it is a traditional knowledge. Now, this is the success of TKDL against biopiracy in last since its inception. Like 241 patents, whether this patent application have either been cancelled, withdrawn, terminated, declared dead, or the claims have been amended by the applicants or have been rejected by examiner on the basis of TKDL proposal. Okay, so this way TKDL is working. Different patent office name has been mentioned over here, and number of patent they have worked. So either they are cancelled or withdrawn by the patentees. And these are the last major suggested changes for Indian herbal drug industry. Okay, so I think these are the references. Yes, ma'am. Sorry.
Okay, thank you very much, madam. And it's a really very nice and informative technical session. And we had learned so many things regarding the regulatory aspects of our India for a herbal drug. Okay. So thank you very much, madam, for this technical nice session. Now, on behalf of Parul University, I want to felicitate you by the e-certificate, and afterward I will mail you. So can I share my screen, ma'am?
Oh, sure, ma'am, and it's fine. You are not sharing. You'll be. You can
No,
send it
ma'am.
me through mail.
No, 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 no. Just 1 minute. Madam, can you see this certificate?
Yes, ma'am. Yes. Thank
Okay.
you very much.
So please
Thank you very much.
accept on behalf of Parun University.
Yes. Thank you, madam.
Thank you, ma'am. Okay. So yes, one more thing is remaining, that if any participant have any question regarding this today's topic, then you can ask to ma'am.
Thank you, Dr. Veena. This is Dr. Professor CPS Verma from Laureate Institute of Pharmacy, Kathok, Jalamukhi, Himachal Pradesh.
Yes.
Ma'am, I
Good
want
afternoon,
to
sir.
ask about the scientific validation of
Yes,
the
sir.
herbal drug as far as healthcare and dietary products and nutraceuticals are concerned. What is your opinion? You have discussed all the parameters or all the methods of evaluation, analytical method and pharmacological, physical, chemical. What is how you can you elaborate
Yeah.
animal model?
Okay, sir. With respect to the nutraceuticals and dietary or functional foods which are trendy in market nowadays, so definitely it should be tested. Okay, these are the nutraceutical products. That means active constituent is present. Okay, so it should be tested biologically, chemically. Biological activities, if it has not been proven, then no one going to accept that thing. Okay, and is not allowed to send in the market also. So it should be tested by through animal models. That means biological activities, and the selection of model that depends on the therapeutic use of that particular product. So what kind of for what kind of disease you are presenting your product, launching your product, so accordingly you need to select the biological model.
Thank you, man. It's a nice this answer. And one more question from regulatory issue. I would like to ask about the Schedule Z of ASU drug and Schedule T about the ASU drugs. Are you satisfied, or you? What is your recommendation if you want the
Sir,
things should be more emphasized or advanced? What is your opinion?
Schedule T and Schedule Z. Okay, so as the time passes, we need to change. We need to make some necessary changes to meet the requirement or to fill the requirements of society. It should be continuously reframed and Right now, we are not having Schedule Z. It has been the Constitution has not still passed for Schedule Z. Okay, so we are just waiting. It has been amended in I think in 2013, but still it has not been passed. So that kind of strict regulation must be there with respect to herbal medicine as well as Ayush medicine in India. So people will rely on us and will be having a huge market as we are having huge resources. We are having very good wealth in terms of plant resources, but still we are very much lag behind to the China. Okay, China is also having wide biodiversity, and if we look the market of China, that is around 3 million or 3 billions, right, dollars. And and as compared to China, we are only 1% of our export is there for herbal medicine. So if we want to increase our this export as well as utilization, if you want to familiarize the internationally with our herbal products, so we need to stringent our regulations.
Okay, ma'am. Thank you. Thank you very much.
Yes, sir.
Uh, yes, anyone wants to ask anything? Okay, okay, fine. So thank you very much, madam, again. And I'm going to share the feedback link in the chat box. So you can find the chat box link.
Thank you, madam.
So,
And again, thank you very much for all the team of Parul University for inviting me. And ma'am, can I leave now?
okay, thank you very much, madam, again. And very nice session is there. So I have learned so many things about the herbal drug and its industry. So thank you very much, madam, for your valuable time. And again, thank you very much from the whole team of Faculty of Pharmacy, Parul University.
Thank you, ma'am. Thank you, everyone.
Okay. So all the participants, you can find the link of feedback form in the chat box, and you can leave the meeting. Now I'm concluding the webinar session, and thank you very much for your valuable time for attending this webinar. Thank you all. Yes, participant, you can leave the meeting, no problem. Thank you all.